Welcome to this video going through the multiple choice questions which I set you on the topic of biochemistry. So that's module 2.2 from the OCR A-level biology syllabus. So first thing is to get these questions and to make sure you've completed them first. I'll set them for you on Microsoft Teams, or you can get them from the website, which I will put the link in the description of the video below. Okay. Let's get going. We're going to mark these, so if you get them right, just tick. If you get them wrong, do make some notes on the page about how you know you've gone wrong, so that you don't make the same mistake again, because they reuse these questions quite a lot in papers. So you want to know these answers and understand why you went wrong first time, so you don't make the same mistake again. Okay, without further ado, let's get going. So this one, first one, solution of, this is important here, sucrose. Sucrose is the thing that they have used and was heated at 80 C for 5 minutes with Benedict's reagent. What was the resulting colour? Now the answer here is blue, and the reason is because sucrose is a non-reducing sugar, and the Benedict's test as carried out here only tests for reducing sugars. To test for non-reducing sugars, you have to add the acid hydrolysis step. I can put that here as well. Acid hydrolysis step would allow it to be detected with the Benedict's test. Okay, second question. Carbon compound extracted from living tissue was found to contain exactly twice as many hydrogen atoms as oxygen atoms. So this means that it's going to be most likely a carbohydrate, because carbohydrate follows this basic formula, sort of CH2O to the N. Okay, so twice the number of hydrogens as carbons. So that answer is A. Question 3, 4, 5, and 6 are all about these statements here. So reduction, oxidation, hydrolysis, and condensation. Let's first sort of define each of those processes. So reduction can be adding hydrogen, or it can be removing oxygen. And it can also be gain of electrons. And oxidation is really the opposite of that. So it's just so it could be subtracting hydrogen, adding oxygen, or losing electrons. So remember, oil rig. Hydrolysis basically means splitting. And condensation basically means joining things together and forming water in the process. So having said that, maltose into glucose. Well, that is a disaccharide. Rub that out. Maltose is a disaccharide, and it consists of 2 glucose molecules. If I were to draw this little hexagon like that, so maltose is 2 of those little hexagons together, and glucose is the 1, but they're separated now, so this is definitely hydrolysis. Amino acids into a polypeptide, well, that's joining monomers into the polymer, and that is a condensation reaction. This, remember, would happen at the ribosome, is where this would happen. Amino acids into a polypeptide, just write that underneath, ribosome. Cytochrome, this is a tough one here. Cytochrome C Fe2 plus to cytochrome C Fe3 plus. Well, we really need to focus in just on this Fe2 plus to Fe3 plus. What is going on there? We are losing 1 electron, and if we're losing an electron, therefore, remember, oil rig, that means we are oxidizing. So the answer for that one is oxidation, which is B. Here, we've got a similar thing going on. We're removing a hydrogen, so that would be an oxidation again. So that's B. So remember, oxidation here, I didn't write it. It's basically the exact opposite of what I said for reduction. Subtracting hydrogens, adding oxygen, or losing electrons. Okay, let's move on. Again, similar style of question. We've got a list of different types of bond, and then we want to know what type of molecule would we find them in. So Glycosidic bonds, let's first look at each bond in turn. This would be found in a carbohydrate. So remember, we can have 1,4 or 1,6 glycosidic bonds found in different carbohydrates. Peptide bonds, well, that's easy. That's found in a polypeptide or a protein. Ester bonds, they are found in triglycerides or phospholipids. So I've given you the answer to one of them already. Now, notice that I didn't say that ester bonds are found in all lipids, because if you want to be really picky, which I do want you to be really picky, there is a lipid called cholesterol which doesn't have an ester bond in it. It's just a molecule that we classify as a lipid, but it's a sort of single molecule and doesn't have an ester bond in it. Okay. Phosphodiester, that is in DNA. Now, I know we haven't looked at DNA on our mind maps yet, but we will do next week. So that's in DNA or RNA. Okay, so I've basically given out your answers here. So hemoglobin, that is, of course, a protein, the protein that carries oxygen around the body, found in red blood cells. Triglycerides, that's our fat stores are made of triglyceride fat molecules. So that is going to be C, ester bond. There'd be 3 ester bonds there between the glycerol and the 3 fatty acids in a triglyceride. Collagen is another protein. So the answer is B. Remember, collagen is a structural protein, as opposed to hemoglobin, which is a transport protein and a globular protein. RNA, that's going to be the phosphodiester bond. Moving on. Which of these compounds would contain the smallest number of oxygen atoms? Well, that should be relatively easy. Actually, I do have to think about it a little bit. Now let's think about it. Okay, so we're almost actually going to draw the structures of these. So a saturated fatty acid. Now, if you think about the end of a fatty acid, it has that carboxylic acid group. So it has it like this. There's an O there, C there, and there's an OH. And then if it's saturated, it's just wiggle, 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 like that. Okay, with this being a carbon and hydrogen backbone. Polypeptide, that's a protein. It's going to have loads of oxygen atoms. So it's definitely not this one. Because remember, in the polypeptide, you've got that peptide bond, which occurs multiple times, which looks like this, and it just goes on and on and on. So there's lots of oxygens in the polypeptide. Polysaccharide, again, that's a carbohydrate. There's loads, hundreds and hundreds of thousands of oxygen atoms. What about glycerol? Okay, glycerol, we actually need to know the structure of it. Let's draw it. I'm going to draw it in red. It is like this. 3 carbons in a chain, 3 OH groups, like that, and then the rest are hydrogens. So actually we've got our answer now. If we compare the saturated fatty acid to glycerol, the saturated fatty acid has 2 oxygen atoms, glycerol has 3. So therefore the answer is A. Okay, general formula for a disaccharide. Well, this is a tricky one. You've got to think about it a little bit. First of all, let's look at glucose. You should know the formula for glucose. C6H12O6. Now, what happens when we make a disaccharide? We have to put 2 of those together, but we have a condensation reaction at the same time. So what we're going to do is we're going to sort of times it by 2 to go to C12H24O12, but then we've got to subtract the water molecule that is produced, the H2O, in which case we're going to get, if you do that maths, you're going to get C12H22 and O11. Ta-da! So therefore the answer is D. Like that one. Okay, 13. Which one of these carbohydrates contains 1 to 6 glycosidic bonds? Okay, well, let's say what we know. Amylose, that's alpha 1,4 only. Amylopectin, that's got alpha 1,4 plus alpha 1,6. So it's most likely going to be that one. Let's just go through each one. Maltose, that's just a disaccharide, and it's alpha 1,4. There's just one alpha 1,4 glycosidic bond in maltose holding the 2 glucoses together. Cellulose, this is a slightly different one. It's a beta 1,4 glycosidic bond. So the only one that has this alpha 1,6 is amylopectin. So the answer is B. 14. On complete hydrolysis, remember that means splitting, which of the following would produce the fewest molecules? That is really interesting. I like this one. Okay, so complete hydrolysis. I have to think through this one. Phospholipid, well, we get the phosphate group. I'll just write letters here. Plus we get the glycerol, which is in the middle. Plus we get 2 times fatty acids. Okay, so therefore we'd have 1, 2, 3, 4 molecules. Triglyceride, we'd have the glycerol plus 3 times fatty acids. So therefore that is going to be 4 again. Sucrose, well, if you split that, you just get 2. That's the glucose and the fructose polypeptide. You get thousands of amino acids. Sucrose here, let's just go into a bit more detail. Sucrose, you've got to remember, is a disaccharide which consists of 1 glucose plus 1 fructose, chemically joined. So the answer is therefore C. It is sucrose. Okay, moving on. Which monosaccharide is shown in the diagram below? Now, the only monosaccharide that you need to be able to draw and recognize the structures of are alpha glucose and beta glucose. So that is pretty much a big clue there straight away. Now, if you didn't recognize the structure, another thing you do need to know is that both ribose and deoxyribose, those are both pentose sugars. Okay, which means they have 5 carbons, whereas both these are hexose sugars. So what is it? How many carbons are there? 1, 2, 3, 4, 5, 6. It's a hexose. And which one is it? This one is beta glucose. There. That is right. Yes, that's right. And that is because the OH on carbon 1, let's highlight it actually, the OH on carbon 1 faces up. I'm going to highlight it green. So it's this bit, this group here, that faces up, which makes it beta glucose. So you can say that it's facing up. Remember, we sort of talk about it with reference to the direction that this carbon 6 is pointing. Okay, so it's facing up out of the plane in the same way as the 6th carbon is. And you can also say it's sort of pointing the opposite way to this one. You just got to remember that. Okay, 16. Amino acids vary according to their R groups. Which one of the following R groups would be hydrophobic? This is getting quite chemical, the knowledge here. So let's look at each one. This one would be pretty hydrophobic. That one not so much. Okay, this would be hydrophilic. So the oxygens, basically, if there's a polar group, it's going to be hydrophilic. So that oxygen makes it hydrophilic. This 2 oxygens here makes it hydrophilic. And H, it's not really hydrophilic or hydrophobic, it's just kind of in the middle. So the answer is the first one. The answer is A. It's the most hydrophobic group. So what notes to make if you didn't quite understand that. So hydrophilic, it basically requires one of a few atoms. So if you have an oxygen in there, tablet's frozen, there we go. If you have an oxygen or if you have a nitrogen, or those are really, yeah, oxygens or nitrogens make it hydrophilic. And if you don't have any oxygens or nitrogens, then it's hydrophobic. That's pretty much it for biology. Can't think of any other atoms that would make something hydrophilic in biology, really. No, that's about it. Okay. 17. Substances which can act both as acids and bases are used in experiments to maintain a constant pH. These substances are known as buffers. Okay, I did briefly mention that in our mind map video. I said that an amino acid has a slight buffering ability because, so let's do an example here. Because an amino acid has its R group, double bond O, OH. Here's a hydrogen, and then over here, oops, we're going to have enough space. We've got the amine group. And remember, an amino acid can act as a buffer because the amine group, let's highlight that in bluish. So the amine group, which is this group here, can act as a base. And on the opposite side, let's try and highlight in a kind of orangey color. On the opposite side, this carboxylic acid group can act as an acid. If you do chemistry, you'll be a bit more familiar with what we mean when we say acids and bases, and that's because this group can lose this hydrogen here, so it can act as a proton donor. This is mainly for the chemists amongst you. And this group here can act as a proton acceptor. So this is again for the chemists. If you don't do chemistry, don't worry about this. But on this nitrogen here, we've actually got a lone pair of electrons. Let's see if I can remember my chemical notation correctly. It's been a while since I did chemistry A level. There's the lone pair. And basically a proton can kind of slide in and dock on that lone pair. So this can act as a proton acceptor, which is a base. That's just going a little bit beyond, and then we put the H there and it would kind of do this, wouldn't it, I think. It's going a little bit beyond the A-level biology syllabus, pushing into the chemistry syllabus to explain how an amino acid can act as a buffer. Okay, moving on to 18, just 3 more questions to go. Which of the following molecules does not yield only alpha glucose molecules on complete hydrolysis? Got to know your carbohydrates here. Well, amylose, that is just a polymer of all alpha glucoses, and remember it's the one that kind of looks a bit like a spiral, like that of alpha glucoses. Glycogen, that is a molecule that is sort of quite branched like that, but it's only alpha glucoses. Remember it's alpha 1,4 and alpha 1,6. Very branched polysaccharide found in animals. Maltose, that is the disaccharide. I drew it earlier. Tried to draw 2 hexagons. Trying to do it again. There we go. Sucrose, that's the one. That's the one we've got to say. Because sucrose, you've got to remember it. I said it earlier. Can you remember what it is? It is a glucose plus a fructose. Really got to know your disaccharides for this part of the syllabus. Okay, 19. We didn't really talk about this in the mind map. It's more to do with experiments within separating molecules, but the description given here, something used to separate molecules according to their differential solubility in a given solvent, is chromatography. So that's B. Remember, you've got your sort of, you basically got your beaker like that. Oops, that beaker's gone a bit wonky. You've got your solvent in the bottom. You've got your piece of chromatography paper just sort of sitting in there. Oops, I'm going to draw that one again. That's awful. You've got your piece of chromatography paper sort of sitting in there. Slightly better. And then you sort of have you have your sort of samples sort of spaced along there. This, you rule a line here with a pencil, not a pen. And then these spots move up the paper. As the solvent moves through the paper, the spots move up. And if a component of this spot here is very soluble in the solvent, then it will move quite rapidly up the top, and it would sort of end up kind of up here maybe. And if there was another component of this spot which was less soluble in the solvent, let's do a different color, then it would sort of not move as rapidly. And then we could compare that with the other blobs, you know, and see which, you know, how those different blobs compared in terms of their contents. Okay, I'll see if I can put a little GIF in on the side to show you that. Last question, question 20. Ooh, bit of maths question here. Okay, tripeptide. It's maths, it's also understanding this word, tripeptide. Tripeptide, tri means 3. So that's 3 amino acids together. So we've got amino acid 1, amino acid 2, and amino acid 3 in a chain. How many different versions or combinations could you have if there are 20 amino acids? Well, you can have 20 different amino acids at the first position. You could have 20 amino acids at the second position. And you could have 20 amino acids at the third position. So the answer is 20 cubed, which is 8,000. So 20 times 20 is 400. Times 20 again is 8,000. So the answer is D. Okay, so that's the first set of biochemistry questions done. So make sure to send me a picture with your mark. Either you can just send me the front page, or you can scan the whole thing to me if you'd like to. But I do want you to make sure that you've made notes on the ones that you've got wrong, because as I said, these questions come up again and again, and you should be familiar with the common types of question they ask, so that if you get this question in an exam, you'll do well. All right, hope that was useful. Thanks very much. Bye-bye.